Hi, everybody, and welcome to our module on connective tissue. Connective tissue is the tissue in our body that supports and connects organs and other structures. And there are a number of components of connective tissue, but the important ones for you to know for the Step 1 exam are collagen, elastin, and fibrillin, and we'll go over all three of these components in this video now. We'll start with collagen. Collagens are a family of fibrous proteins, and they are the most abundant proteins in the human body. Of the entire mass of proteins in the body, 25% is made up of collagens, and collagens are synthesized and secreted by connective tissue cells into the connective tissue. Collagen is made up of three long protein chains called alpha chains, and the basic unit of a collagen molecule is called the triple helix. This is a picture of one shown on the screen here. It's three long alpha chains linked together. And there are 42 different genes that have been described for alpha chains, and different combinations of alpha chain proteins give different collagen types to make up all the different types of collagen found in the body. Some are very stiff, some are more stretchy and flexible. They are all collagens, they just have different alpha chains. Collagen contains large amounts of three amino acids, proline, lysine, and glycine. These are their structures shown on the screen here. And it's often made up of repeating units of a glycine amino acid followed by two others. The two others may be proline, or they could be lysine, or they could be some other amino acid. So what this means is much of the collagen is made up of glycine, less of it is made up of other amino acids, but among those other amino acids, proline and lysine are very common. As I said before, there are many different types of collagens determined by the different alpha chains that make up the triple helix. And the collagens are numbered type 1, type 2, type 3, and so on. And let's start by talking about type 1 collagen, which is the most common form. This makes up 90% of collagen in the body. This is found in a number of locations, including bone, and I've bolded the word bone because it's helpful if you remember that type 1 collagen is important for bone, because later we'll talk about a rare condition called osteogenesis imperfecta, which is also called brittle bone disease. And this condition results from abnormal formation of type 1 collagen, and it leads to frequent fractures. Other locations for type 1 collagen include the skin, the tendons, and ligaments. You can find it in the cornea of the eye. You can also find it in some internal organs. Type 2 collagen is found in cartilage. You can also find it in the intervertebral discs. It also is a component of the vitreous humor found in the eye. Type 3 collagen is found in the skin, just like type 1 collagen. It's also found in blood vessels. And this form of collagen is abnormal in some of the subtypes of Ehlers Danlos Syndrome, which we'll talk about at the end of this video. And you may read in your textbooks about so-called fibrillar collagens. These are the type 1, 2, and 3 collagens. These are called fibrillar because the collagen molecules in these types of collagen assemble into polymers called fibrils, which we'll talk about in a minute. There are also many types of non-fibrillar collagen, but we're only going to talk about one because this is the one that sometimes comes up on the boards, and that's type 4 collagen. The reason this type of collagen is important is because it's found in basement membrane. So you can find it in the basal lamina, beneath the epithelial layer in many structures. You can also find it in the lens of the eye and in the cochlea of the ear. And if you understand those locations for type 4 collagen, then it's very easy to understand the rare genetic disorder Alport syndrome, which is also called hereditary nephritis. This condition is caused by a genetic type 4 collagen defect. It's caused by mutations in the genes that code for the alpha 3, alpha 4, or alpha 5 chains of type 4 collagen. It's most commonly X-linked, so in a board question, it's likely to be a boy child, and the classic triad is hematuria from loss of the basement membranes in the glomeruli, hearing loss from loss of type 4 collagen in the cochlea, and ocular disturbances from loss of type 4 collagen in the eye. Now let's talk about how collagen is synthesized. And the first thing you should know is that collagen undergoes extensive post-translational modification. So once the proteins are translated from mRNA, that is only the beginning of the process. There's then a long process of modifying the proteins and amino acids until collagen assumes its final form in connective tissue. The process begins by synthesis of alpha chains on the ribosomes in the rough endoplasmic reticulum. These alpha chains contain signal molecules, and they are referred to sometimes as pre-pro collagen. The signal molecules direct the alpha chains into the lumen of the endoplasmic reticulum, and at this point they are referred to as pro-alpha chains. In the endoplasmic reticulum, there are a number of modifications made to those alpha chains. First of all, some of the proline and lysine amino acids are hydroxylated. They then become what's known as hydroxyproline or hydroxylysine. This process requires vitamin C as a cofactor. It's a cofactor for the hydroxylase enzymes. And therefore, if you are deficient in vitamin C, you will have problems synthesizing collagen, and this leads to the condition of scurvy, which we'll talk about in a minute. Other modifications that occur in the endoplasmic reticulum include the fact that some of the hydroxylysines are glycosylated, meaning that sugar molecules are added to the hydroxylysine amino acids. Shown on the screen are the chemical reactions that occur in the process of hydroxylation in the endoplasmic reticulum. So lysine and proline amino acids found in the pro-alpha chains undergo hydroxylation, which requires vitamin C. Lysine becomes hydroxylysine, which has a hydroxyl group not present in lysine. Proline becomes hydroxyproline, which also has a hydroxyl group present that was not present in proline. And shown on this slide is the process of glycosylation. The hydroxylysine amino acids that were formed by the process on the last slide can be glycosylated so that a sugar molecule, often a glucose, but sometimes another sugar, is added to the hydroxylysine, and it is then said to be glycosylated. Scurvy is the clinical condition that results from vitamin C deficiency. It's caused because of defective formation of pro-alpha chains because there is no vitamin C around to hydroxylate the pro-alpha chains. The alpha chains that are formed will not form a triple helix. They will be degraded in cells and not secreted. And the first place this will become evident is in the blood vessels. The blood vessels need normal formation of collagen. So if you can't form collagen because of a vitamin C deficiency, the first thing you'll notice is bleeding or bruising. Classically, it occurs in the gums, as shown in this picture here. There can be bleeding in the gums. In addition, people can lose their teeth, and there can be poor wound healing. And all of this is the result of vitamin C deficiency and inability to hydroxylate those proline and lysine residues in pro-alpha chains. Back to our discussion of collagen synthesis. In the endoplasmic reticulum, some of the pro-alpha chains have molecules attached to their end called propeptides. Propeptides are extra amino acids, that's why they're called peptides, that are attached to the N and C ends of the pro-alpha chains. Remember that proteins like pro-alpha chains have one end that is an amine group, that's the N end, and another end that is a carboxyl group, that's the C end. And both of these ends have propeptides attached in some of the pro-alpha chains. This occurs in any of the pro-alpha chains that will go on to form fibrillar collagens like type 1, 2, and 3. And the reason these propeptides are important is because they form disulfide bonds that help stabilize alpha chains. At the bottom of the screen is a picture of a disulfide bond to help you remember this. At this point, 3 pro-alpha chains can combine, and when they do, the structure they form is called procollagen. This is the triple helix formation of 3 pro-alpha chains. After procollagen forms, it moves through the Golgi apparatus, and then it is excreted from cells by exocytosis. After it leaves the cells, the propeptides at both ends are cleaved, and once those propeptides are removed, it is no longer called procollagen, it is called tropocollagen. Tropocollagen is individual triple helix alpha chain molecules with no propeptides. They have been removed, and it is not yet cross-linked. That is the next step that must occur in forming collagen. So tropocollagen will go on to form collagen fibrils. These form because tropocollagen is much less soluble than procollagen, so it will fall out of solution and fibrils will begin to self-assemble. They are strengthened by lysine cross-linking. I'll show you a picture of this on the next slide. There is an extracellular enzyme called lysyl oxidase that can cross-link lysine in collagen fibrils. This requires copper as a cofactor. There's a picture of copper on the screen to remind you of this. And then once these collagen fibrils have been formed, they can go on to link together. Collagen fibers, not fibrils, fibers, are bundles of triple helices. They are bundles of collagen fibrils linked together. Here's a picture to help you better understand how cross-linking works. Tropocollagen is a triple helix. It contains lysine residues which have NH3 groups attached. Lysyl oxidase will oxidize these residues and add an oxygen group. Then 2 oxidized residues can link together, and thus you will have cross-linked tropocollagen found in collagen fibrils. So here's a summary slide to help you remember how this whole process works. Most of the action takes place in the endoplasmic reticulum or the extracellular space. Pro-alpha chains are found in the lumen of the endoplasmic reticulum. They undergo hydroxylation and glycosylation. They then link together in a triple helix to be called procollagen with propeptides on both ends. These molecules are excreted by the cell and the propeptides are removed. The structure is then called tropocollagen, which has no propeptides. This then undergoes cross-linking and forms collagen fibrils. So now let's talk about some of the clinical disorders that are related to problems with collagen. First of all, the wrinkles that we develop as we age are related to decreased production of collagen and also elastin, which we'll talk about later, in the dermis layer of the skin. In addition, collagen and elastin fibers tend to thicken and clump as we age, and that's what gives the wrinkling of the skin. In the immunology section, I have a video on scleroderma, which is also called systemic sclerosis. This is an autoimmune disorder that results in stiff, hardened tissue. That's why it's called sclerosis. The skin is often involved, and so are many other organ systems. And this disorder is caused by fibroblast activation that leads to excess collagen deposition in skin and other organs. Osteogenesis imperfecta is also called brittle bone disease. This is a family of genetic bone disorders caused by mutations in genes that are necessary for collagen formation. There's a range of severity. Some of the forms are even lethal in utero, but they all involve osteoporosis and fractures, and they're all due to defective or deficient collagen production. The type 1 form of osteogenesis imperfecta is the most common form. It can run in families in an autosomal dominant fashion, and it is due to a genetic defect in either the COL1A1 or COL1A2 genes. And COL1 means type 1 collagen, so that can help remind you that the problem in this disorder is genes that encode for the alpha chains of type 1 collagen. In this form of osteogenesis imperfecta, there are abnormal or absent alpha chains for type 1 collagen, and the triple helix is not formed normally. The result is decreased production of type 1 collagen. The other forms are much less common. The type 2 form is lethal in utero. Types 3 and 4 are more severe than type 1, and thus there is a range of severity, with type 2 being the most severe, type 1 being the least severe, and type 3 and 4 being in the middle. Let's talk about the clinical features of osteogenesis imperfecta. Children with this disorder have multiple recurrent fractures with minimal trauma. It is often confused with child abuse until the disorder is diagnosed. Another classic feature is blue sclera. This is a picture of blue sclera on the screen here. The sclera should be white, but they have a blue hue to them in osteogenesis imperfecta. This is because of clear connective tissue in this disorder overlying the veins in the sclera. In addition, these children often have hearing loss. This is because, remember, there are important bones in the ear, the malleus, the incus, and the stapes. These are called the ossicles, and they are abnormal in osteogenesis imperfecta. Some people with OI have what's known as dentinogenesis imperfecta. This is rarely seen in the type 1 or more common form of the disorder. It's common in types 3 and 4. People with dentinogenesis imperfecta have discolored teeth. They're often blue-gray or yellow-brown. This is a picture of someone with this problem on the screen here. The teeth also are translucent or shiny, and they can be weak and easily fall out or break. And then, as you can imagine, if the bones are abnormal, these children often have bony deformities, especially in some of the rarer forms of this disorder, and they can grow to have a short stature. Another genetic disorder related to collagen formation is Ehlers Danlos Syndrome. Just like osteogenesis imperfecta, this is a family of conditions. They're all genetic connective tissue disorders. There's a range of severity and a range of inheritance patterns, but they're all caused by defective collagen synthesis. And in contrast to osteogenesis imperfecta, the collagen problems in Ehlers Danlos primarily affect the joints and the skin rather than the bones. There are many subtypes of Ehlers Danlos syndrome, just like there are many subtypes of osteogenesis imperfecta. The two we're going to talk about in this video are the classic type and the vascular type. The classic type is often inherited in an autosomal dominant fashion, although it's often a de novo mutation, meaning that a parent may not have the condition when a child develops this. However, that child can pass it on to his or her offspring in an autosomal dominant fashion. This type is caused by defects in the COL5A1 or COL5A2 genes, which code for type 5 collagen. And remember, COL5 tells you which type of collagen the gene codes for. And type 5 collagen we haven't talked about before, but it interacts with other collagens. The vascular type can also be passed through families in an autosomal dominant fashion. It is caused by problems in the COL3A1 gene, which codes for type 3 collagen. And this is called the vascular type because it's found in blood vessels. It's also found in the skin. Let's talk about some of the clinical features that can be seen in the classic type of Ehlers Danlos syndrome. This is the type related to type 5 collagen. First of all, these patients often have joint hypermobility. Shown at the bottom left of the screen is a picture of a patient with Ehlers Danlos syndrome. You can see that this patient is able to bend their finger all the way back. This is evidence of joint hypermobility. Another classic feature is hyperextensible skin. At the bottom right side of the screen, you can see this person is able to pull their skin entirely off of their leg. This can be seen in Ehlers Danlos syndrome. In addition, the skin is classically described as having a velvety appearance. Patients with the classic type of Ehlers Danlos syndrome can have easy bruising. They can also develop thin, wide scars that are referred to classically as cigarette paper scars. These patients can have mitral valve prolapse. Remember, in mitral valve prolapse, The mitral valve is stretchy and it billows into the left atrium, and that can be seen in this condition. And then just an aside here, but many of these features are also seen in some of the other subtypes to varying degrees. Some of them have joint hypermobility, some have skin findings, some have a bit of both. Now let's focus on the vascular type. This is the type of Ehlers Danlos Syndrome related to type 3 collagen. These patients can have thin skin and easy bruising, and these symptoms are somewhat similar to the classic type. But what's unique about the vascular type is that they have problems with large arteries, especially they can develop rupture of large arteries in the brain. They can develop CNS or berry aneurysms, which I discuss in the neurology section. This is a picture of one shown on the screen here, and these can rupture and these can potentially be a cause of death. In addition, they can have rupture of what's known as hollow organs. This includes intestinal perforation or rupture of the uterus during pregnancy. And because of these problems with rupture of arteries and rupture of organs, this is a life-threatening form of EDS. This is the worst form to have. 80% of patients with this form of Ehlers-Danlos have a vascular event or rupture by 40 years of age, and the median age of death is only 48 years old. And then finally, there is a very rare disorder called Menkes disease, which can lead to problems with collagen formation. This is an X-linked recessive disorder caused by mutations in a gene called ATP7A. This gene gets its name because it codes for a protein that is an ATPase involved in intestinal copper uptake and transport. So people with a mutation of this gene have impaired copper absorption, and this leads to deficiency. And just an aside here, in the gastroenterology section, I have a video on Wilson's disease. Wilson's disease is caused by copper excess. The gene that causes Wilson's disease is called ATP7B, and it is closely related to the ATP7A gene that causes Menkes disease. In fact, you can think of Menkes disease as the opposite of Wilson's disease. Wilson's is copper excess, this is copper deficiency. And the reason this is related to collagen formation is that, like I told you before, lysyl oxidase requires copper as a cofactor. So in the state of copper deficiency, you will have poor lysyl oxidase activity. This means you won't be able to cross-link collagen. The classic feature of this condition, and the one you need to remember in order to recognize it in a board question, is that it will describe a male child with sparse, brittle, or kinky hair. This is a classic feature. This is a picture of a child with Menkes disease shown in the screen here. In addition, these children tend to have low body temperature, and they have central nervous system features like hypotonia and seizures. They display poor growth and developmental delay. Because of the collagen problems, they have osteoporosis and fractures, and this is usually fatal in childhood. Now let's move on from collagen and talk about elastin, which is another connective tissue protein, and it's the main component of elastic fibers that allows stretching or recoil of connective tissues. Elastin is a very important component of our arteries. It's the dominant elastic protein found in arteries, and it makes up 50% of the aorta. You can also find elastin in the skin, in the lungs, and remember the lungs because we'll talk later about an elastin disorder where there are problems in the lungs. You can also find elastin in ligaments, in the vocal cords, and in the ligamentum flava, which are ligaments you can find in the spine. Elastin contains the amino acids glycine, lysine, and proline, just like collagen. What's different is that the amino acids are mostly non-hydroxylated. There is no hydroxylysine. There's a little bit of hydroxyproline, but much less than collagen, and none of the amino acids are glycosylated. Elastin is secreted by cells as tropoelastin, just like collagen is secreted as tropocollagen, and then it's assembled into elastin fibers with cross-linking in a process somewhat similar to collagen. There are a couple of disorders related to elastin that you should be familiar with. The first one is alpha-1 antitrypsin deficiency. I discussed this condition in detail in the obstructive lung disease video in the pulmonary section. I'll just briefly review it here. It's a genetic disorder inherited in an autosomal codominant fashion. It's codominant, meaning that both alleles for alpha-1 antitrypsin contribute to the phenotype. It's caused by decreased or dysfunctional alpha-1 antitrypsin protein, and this is an important protein for inhibiting the enzyme elastase. Elastase can break down elastin, so when you're missing alpha-1 antitrypsin, there is excessive elastase activity, and that leads to excessive breakdown of elastin. And one of the key features is emphysema. Remember I told you before that elastin is found in the lungs. So patients with this disorder have lung damage just like smokers do, but they usually get it at a younger age, and they can even get it if they are non-smokers. And this is because there's an imbalance between elastase found in neutrophils, which destroys elastin, and the elastase inhibitor alpha-1 antitrypsin, which is supposed to protect elastin. In the genetics video on deletion syndromes, I talk about the rare condition Williams syndrome, which is also called Williams-Barré syndrome. This is caused by a partial deletion of the long arm of chromosome 7, and the deleted portion includes the gene for elastin. Children with this condition classically have an elfin appearance and intellectual disability. I discuss this in greater detail in the video on deletion syndromes, but what you should know for our purposes here is that another classic feature of Williams syndrome is supravalvular aortic stenosis. This is constriction of the ascending aorta above the aortic valve. This is very rare. And remember what I told you before, elastin is a very important component of the aorta. So when it's missing, what you get is constriction of the aorta above the aortic valve. And there's a very high prevalence of this rare condition, supravalvular aortic stenosis, among children with Williams syndrome. And on histologic examination of their aortic tissue, what you see is loss of elastin. The last connective tissue substance we'll discuss is fibrillin. Fibrillin is a glycoprotein, and it is the major component of microfibrils. Microfibrils are a sheath that surrounds an elastin core, and they are a major component of elastic fibers. Elastic fibers are made up of elastins surrounded by microfibrils and some other molecules, and they are abundant in the aorta. And the one thing you want to know about the molecule fibrillin is that when it is deficient, that leads to Marfan syndrome. Marfan syndrome is a genetic connective tissue disorder that results from abnormal fibrillin glycoproteins. It is caused by mutations of the FBN1 gene on chromosome 15, which codes for fibrillin 1, and it has clinical manifestations that involve the bones, joints, heart, and eyes. The classic appearance of a patient with Marfan syndrome is tall with a long wingspan. There is a theory that Abraham Lincoln, President of the United States during the Civil War, had Marfan syndrome, and if you remember what he looks like, it's easy to remember the phenotype of Marfan syndrome. The problem here is different from osteogenesis imperfecta, where the bones are abnormal. The issue in Marfan syndrome is that the connective tissue is abnormal, and it allows the bones to grow excessively long, and thus these patients are very tall and they have long arms and therefore a long wingspan. Another classic finding is pectus excavatum, or a sunken chest. This is a picture of what that looks like on the screen here. In the extremities, patients with Marfan syndrome have a number of classic findings. They have hypermobile joints that share some similarities with Ehlers Danlos. They tend to have long fingers and toes, and they are often said to have arachnodactylity. This means long, curved fingers. It gets the name arachno because it looks like a spider's legs. There are two classic signs in the extremities. One's called the wrist sign. I'll show you a picture of this in a minute. This is where the tip of the thumb covers the entire fingernail of the fifth finger. And then the thumb sign is where the thumb protrudes beyond the ulnar border. Here's the thumb sign on the left side of the screen. If you try to do this with your own hand, assuming you don't have Marfan syndrome, you won't be able to stick your thumb out that far. And this is the wrist sign on the right side of the screen. If you try to do this, you shouldn't be able to wrap your fingers completely around your wrist, but people with Marfan syndrome can do this. There are a couple of eye findings in patients with Marfan syndrome. They can develop cataracts at an early age. Most people who develop cataracts are elderly, but in the case of Marfan syndrome, the cataracts are said to be pre-senile because they develop at an early age. In addition, they can develop dislocation of the lens. Now, dislocation of the lens commonly occurs due to trauma, but sometimes it happens in association with a systemic condition. And the most common systemic condition associated with dislocation of the lens is Marfan syndrome. This is a picture of a dislocated lens shown in the screen here. Classically, the type of dislocation that occurs in Marfan syndrome is the lens dislocated upward and outward. And I talk about this in more detail in the eye videos in the neurology section. And then finally, there are a couple of cardiovascular manifestations of Marfan syndrome. They can develop mitral valve prolapse. Their mitral valve can be stretchy, just like patients have with Ehlers Danlos syndrome. And then the most dangerous complication of Marfan syndrome is that these patients are at high risk for aortic aneurysms and dissection. Remember that elastin and the fibrillin that surrounds it is very prevalent in the aorta. So these people have weak aortas. They can develop what's known as cystic medial necrosis. This means the presence of cysts and necrosis in the medial layer of their aorta, and that predisposes to aortic aneurysms and aortic dissection. And the last thing I'll mention is you will sometimes hear that certain conditions are associated with a Marfanoid habitus. What people mean when they say this is that patients with these conditions look like they could have Marfan syndrome. So they are tall with a long wingspan and they have long fingers. And this is seen in some rare systemic diseases. You can see a Marfanoid habitus in homocystinuria. You can see it in multiple endocrine neoplasia type 2B. And you can also see it in some of the rare forms of Ehlers Danlos syndrome. And that concludes our video on connective tissue.